So, good morning. And I hope everybody will answer my questions with as much volume as Anissa was able to get out of you the second time. So, today, I gave the title, Your DNA, Sense or Nonsense. We're going to talk about RNA as well as DNA, but we're going to start out with DNA. So, how many people know what DNA is? Okay, does somebody want to tell me what DNA is? What's DNA?
Deoxyribonucleic acid.
Fantastic. What does it do?
It's basically like the genetic code for all life.
Okay, and is it an atom or is it a molecule? You guys are really smart. But it's an unusual molecule in that it's very, very long and very thin. So you can think of it as being like a piece of string. This would probably be equivalent to the amount of DNA that we have in one of our chromosomes. If I were to spool the entire piece of thread, and there's lots and lots and lots of it, but it's pretty skinny, right? And you probably all know that it has 4 bases, and that the code for our proteins is in the sequence of those 4 bases along the DNA strands. Okay. How many of you have seen DNA? A couple people have seen DNA. Okay, what I have here, for those of you who haven't, is a solution that's got some DNA in it. You see a sort of gray color, right?
Okay.
But do you see anything else? Okay, and I'm going to put a stirring rod in it, and I'm going to stir it. And sort of nothing happens, right? Now I'm going to add some ethanol. Carefully. Hope I can do this right. Be very disappointing if it doesn't work. Okay, I've got to sort of measure it out. So I've got to put in twice as much ethanol. I'm at 200, so I've got to go up to 600. So I'm going to do this very carefully. Some of it's Sorry it's taking so long here. Okay, so can some of the people who are in the front row come up and tell me what they see now? Come on. Okay. Sort of looks like 2 layers, right?
The bubbles are being held up by strands of DNA. Or of something.
Of something that looks like sort of strands, right? Can people in the back see this at all? Can you see that there are 2 layers? And that there's starting to be something sort of coming out of the layer? Okay, so now I'm going to go in with a stirring rod and do this. And I want other people from the front row to come up and tell me what they're seeing now, so that all that fluffy stuff is starting to get picked up on this rod. See?
It's like spaghetti.
It's like spaghetti, exactly. Yeah, okay. And so what we're seeing here are the long strands of DNA. So can you all see it? Yeah. Now there are long strands of DNA here. Okay, so now you've all seen DNA. And if anybody wants to come up later, I've got more test tubes and more solution if you want to do it yourself afterwards. You can all come up and do this and see the DNA come out a solution. Okay. So let's get back to what we're going to talk about. So everybody already knows what DNA is. So let me talk a little bit about me and my relationship to DNA. You guys learned about DNA in grade school or in junior high school or maybe high school. When I was there, DNA wasn't even mentioned. Even though the double-stranded structure of DNA that you all probably know about, the two strands come apart and then serve as templates for making additional strand so that the information can be passed on to the next generation. You all know this. That had been discovered before I was in high school, but it was so new it hadn't made it to any of the textbooks yet. As a matter of fact, I went back and looked at my high school biology book, and it had one sentence in it about DNA. And you were absolutely right, except what my sentence said was, it is thought that genes might be made of a substance called desoxyribonucleic acid. That was the old term for deoxyribonucleic acid, and that was absolutely it. And I knew I was interested in science, so I majored in science when I got to college. And the time I found out about DNA was like 8 or 9 years after 1953, after the discovery of the double-stranded structure of DNA by Watson and Crick. And the way I found out about it was I happened to get a job at MIT and went to a lab that was in this new field of molecular biology. And they were, of course, studying DNA. And I remember when I got there that I was, and found out about this, that I was absolutely fascinated because I'd known I was interested in science. I thought genetics were really interesting, but I didn't think that anybody would ever be able to put molecules together with life. And when you saw that beautiful double-stranded structure and saw how it could come apart and replicate, you knew immediately that there was a possibility. So I got very excited. But nonetheless, I decided that I should go to medical school, not go to graduate school, and try to study DNA and how DNA worked. And that was because after high school, I never had a woman professor in science. I worked in a number of labs. I never saw a woman professor at any other university, but I did know some women doctors. So I decided that I should go to medical school. And what changed my mind was that the summer before I was about to go to medical school, I ended up working in the lab of a very famous scientist named Joe Gall, who at that point was at the University of Minnesota and then spent most of his career here at Yale in Klein Tower, right next door. And for the first time, he gave me my own project to work on. And I got so excited by doing my own project, instead of helping other people do theirs, which I'd done previously, that I decided I didn't really care whether I would never get to be a professor. All I wanted to do was to make the discoveries and have fun finding out about how DNA and RNA worked. So I went off to graduate school. And that was a time when this field of molecular biology, which now you all know about, was really, really new. And the people in the field thought that if you were going to find out anything, you had to start very simple. And this was actually a good judgment at that time. So they decided that the only things that were worth working on were bacteria, little cells that swim around, and the viruses that infect bacteria. And as a matter of fact, the feeling was so strong that you had to do work with simple systems, that people who were trying to work on complicated cells, like the ones that come out of our bodies, were even sort of scorned as wasting their time because it was so complicated they would never figure anything out. So starting there and looking at what's happened is something I never could have imagined would happen. That we would know, and you guys know this, right? That we would know the 4 billion base pairs of the human genome and the sequence and how it specifies many of the proteins. Or that knowing about DNA would do things like give rise to diagnostics in medicine, therapies in medicine, even DNA forensics. Everybody knows about how DNA is used to find out whether it was this person or that person who was at a crime scene. That all was totally unimaginable when I started. Or that there'd be a multi-billion-dollar biotech industry making lots of money off this whole field. And of course, it was a surprise, and I'll come back to this at the very end, that there were, at that point, that there were no women in science. And today, they're both women science professors. You've just seen 2 of them. And even women as presidents of universities. So that's changed a lot. And I want to say a little bit at the end about women in science. So I've told you now about where I started. And now I want to go on and talk about relating my title, Sense or Nonsense, which is in our DNA, back to something about RNA and something called SNRPs, which are little particles in cells that are very important to the cell for getting the information out of the DNA so that it can be translated into proteins. So let me go on here. Okay, so I'm going to tell you the story of SNRPs. And this is a sort of unusual story in science because a lot of us think of scientists as going in and making discoveries, and then somebody comes along and figures out how to apply them to medicine. But this is a story where a disease called lupus provided the materials with which to find out about the basic science. So do any of you know anybody that has this disease called lupus? A number of hands went up. Any of the kids? You do. So this is a disease that it's about right that a number of people should know. There are about 1 in 1,000 people in our population has lupus. It's more common in women than in men. It's more common in blacks than in whites. There are a number of other diseases that are related to lupus. They're all called autoimmune diseases, and I'll tell you why that is in just a moment. So the other diseases are things like mixed connective tissue disease, dermatomyositis, sometimes arthritis, and scleroderma, which got a lot of publicity about 10 years ago because breast implants were tied up with scleroderma. And scleroderma, it's interesting, is a disease where the sort of outer surfaces of the skin get very hardened. And it's probably the disease that Lot's wife in the Bible had when she turned into a pillar of salt, very stiff. We don't know that, of course, but that's probably what was going on. Okay, so what's happening in people with lupus or these other autoimmune diseases? So how many of you know about antibodies? Lots of kids know about antibodies. What do antibodies do? Over here, antibodies, you want to say? What do antibodies do? It's right there on the slide. You can read it. Yeah.
Antibodies, like when you get sick.
Yeah, when you get infected with a bacteria or a virus.
After you get sick, you're not sick anymore. But if you get sick that same virus again, then the antibodies.
Yeah, right.
They already know what that sickness is.
Great. Okay, so antibodies are something that your body, your immune system makes when it's exposed to something foreign, and that interaction between the antibodies and the foreign molecules clear them from your system so that Next time, you won't get sick. It's exactly what you said. Great. But sometimes in some people, and this is what happens with lupus and these other diseases, something goes wrong with their immune system's ability to tell the difference between molecules that are in your own cells and these molecules that are in bacteria and viruses that are foreign. And so what those people then make, and it's a rare disorder, is antibodies against their own cellular components, which are called autoantibodies. And that's why these are known as autoimmune diseases. And what happens if you have autoantibodies, let's think about it. If they're antibodies against your own cellular components, then let's say you go out in the sunshine and you get a sunburn. That kills your cells, or not a lot, but some of your skin cells, and they die and they dump their contents into your blood. So if you have autoantibodies, then you get these interactions between your antibodies, your autoantibodies, and your own cellular components to form what are called immune complexes. And these things build up in big networks, sort of like we saw with the DNA in your blood stream. And the reason that the disease is called lupus is people get a red rash, and that's because a lot of these complexes have deposited in the little capillaries that are under the skin. Okay, the complexes also deposit in the hair follicles, so people's hair falls out. They deposit in the joints, so people can't move very well. They deposit actually in all the internal organs. These are systemic diseases and very, very difficult to treat. And a lot of people end up succumbing to kidney disease because the immune complexes deposit preferentially in the kidneys, and those are actually antibodies against DNA. So that's the next question, is what is it that in these diseases people make autoantibodies against? And it turns out, for reasons we don't understand, that they're some of the most abundant and highly conserved things inside cells. Okay, how many of you kids have heard that DNA makes RNA and RNA makes protein? Very large fraction of who's here. Great. Okay, so here's a picture of DNA. This is a strand of DNA. There are polymerases moving along making RNA, so strands of RNA, and those are getting coated with ribosomes, which are the protein-making machinery inside the cells. So here you're seeing DNA makes RNA makes protein. And in fact, some patients with lupus make antibodies against ribosomes, protein synthesis factories. Many people with lupus make antibodies against their own DNA, and that's what deposits in the kidneys and causes the real problems. But many also make antibodies against SNRPs. Now, SNRPs aren't in this picture because this is a picture of what goes on in bacteria. And bacteria don't have SNRPs, but our cells have SNRPs. So what are SNRPs, and why do we have them? And this has to do with the sense and the nonsense in our genes. So let me move on to how did I get into this whole area. Here we're looking at DNA, right? And we're looking at the amount of DNA. And you can tell how much there is by, what a big pile of strands there are there. And after this very time when everybody thought you had to be very simple, some molecular biologists started thinking, well, we better start looking at higher cells. And most people said, oh, they'll be just the same. But some people said maybe they'll be different. And we now know they're very different in terms of our DNA. So what this slide shows is here's a virus, and it has about 200 genes, and that's the amount of DNA. Here's a bacterium. It has about 2,000 genes. So does that look like about 10 times as much DNA? Yep. This is a mammalian cell, a huge amount of DNA. And people used to think we had about 100,000 genes. Do any of the kids know how many genes we think we have now? What? Billions? No? Anybody know? Anybody else know? We have probably about 27,000 genes. Anyhow, this amount of DNA was way more than what we would need for 27,000 genes, about 1,000 times more. And this was very puzzling to people. It was also true that mammalian cells made lots and lots of RNA, copying the DNA, but that lots of that RNA got degraded in our cells and never turned into a messenger RNA to make protein. So I got interested in trying to solve that problem on a sabbatical. And the project didn't work, so I gave up and did something else. And when I came back to Yale, everybody in my lab wanted to work on this particular problem. And what had happened in the meanwhile was that it had been discovered that our genes really are different from the genes in bacteria, in that they have pieces in them that are the sense parts, but in between are nonsense bits. And often the nonsense bits are longer than the sense bits. And what our cells need to do that bacteria don't need to do is something called splicing. They need to make cuts at the ends of the sense bits and join them back together to make a messenger RNA that can make protein. And that's illustrated, I think, even better in this slide, where it says, even though the 3-letter code remains, and then there's a bunch of gibberish, that's the intron. And then we go back to the exon, that's the expressed part, and another intron of gibberish, nonsense, and then back to the sense part. So this is what our genes look like. They're sort of genes in pieces. They're separated by these bits of nonsense. So what this meant is that our cells, in contrast to bacterial cells, have to have some machinery in them, right, that's going to recognize these junctions between the nonsense and the sense and cut precisely and join everything back together so that you can make a sense messenger RNA. Yeah?
That's why it's called sense or nonsense.
Exactly. So here's the slide, okay? You know which the sense parts are, and there's the nonsense. And virtually all of our genes, not all of them, but 99% of our genes look like this, okay? So as I mentioned, people in my lab wanted to work on this, but we didn't really know how to do this or what to do. And then came a piece of serendipity. How many kids know what serendipity means? Who wants to? There's a hand up way back there. What's serendipity? Luck. It's when something happens that you weren't expecting. It's not connected to anything, but it turns out to make a difference, usually. And that serendipity was an article that appeared in the journal Nature, and it mentioned patients with MCTD. And I told you before that that was like lupus. It was one of these autoimmune diseases. And what I'd been trying to think about that hadn't worked was the fact that there might be particles in cells and that these particles might have something to do with how the cells got rid of the nonsense. And the minute that we saw this article, people had mentioned to me that, maybe there were patients that made antibodies against what I was trying to work on, but I hadn't known who to contact. When this article came out, I was back at Yale, and I had a MD PhD student in my lab named Michael Lerner. I'll show you his picture later. And I said to Michael, do you know anybody here at Yale who might have patients with MCTD? And he said, sure, I'll go across the street and see Hardin, who was the head of the rheumatology section in the Department of Internal Medicine. And he did that and came back and started to work with these patients' blood that had the autoantibodies in them right away. And eventually what we figured out was that these autoantibodies were directed against cellular components that happened to be SNRPs And what those are little particles that have RNA and protein in them. And those are the things that recognize those junctions between the sense and the nonsense. And what they then do is they get those two ends together and arrange for the nonsense in between to be cut out and the sense parts to be joined back together. And here's a picture of this happening. Here's the DNA again. Here's the RNA. Here are the SNRPs and other things at these what are called splice sites, the ends of the nonsense. And you see these particles building up. Here you see what's called a spliceosome. And here the whole nonsense bit, the intron, is looped out, and it's about to get cut and rejoined and made shorter, and you've gotten rid of the nonsense. Okay? So that's what's going on in your cells all the time. Has to happen virtually any time a gene is turned on, it makes RNA, but that RNA has got introns in it, and it's got to get spliced by this process. So let's think about what that means. Okay, so what it means is that our cells have this extra step in making proteins. They make the RNA, but it's got nonsense in it, and the nonsense has to come out by the SNRPs finding the junctions and making a spliceosome, and then making a messenger RNA. So let me show you, I think, okay. So thinking about this, and now moving forward, this was a number of years ago, what does this mean for our understanding, and what does it mean for disease? Virtually all of our genes have this exon, intron, exon structure, sense, nonsense, etc. And what that means is that there are different ways of splicing the RNA coming off a gene so that you can either make a protein that has the red bit or a protein that doesn't have the red bit. They're different proteins. And turns out that Drosophila has about the same number of genes, the little fruit fly, as we do. But we do more of this stuff, this alternative splicing, so we can get more out of that same number of genes and make more proteins and be more complicated. So that's important in terms of understanding evolution. The other thing is in terms of understanding disease. There are a lot of diseases where the splicing process goes awry in a particular gene. And there are diseases like cystic fibrosis and spinal muscular atrophy, muscular dystrophy, where people are now trying to figure out how to correct the splicing process in those genes. And some progress is being made because what you need to do is get out of it something that can actually, a messenger RNA that can make protein. And I'm just coming up on the end here. This is a picture. This is Michael Lerner, the MD PhD student that started working with the antibodies. Here we were conferring with Sid Altman in the biology department here that day about possible collaborative experiments. So there were a lot of people involved, and one of the really fun things about doing science is that you get to work with a lot of other people, figuring out how to do experiments that tell you how things work. And so what I want to end with is to go back to my thesis advisor, Jim Watson, who's shown there, of Watson and Crick. And these were two other graduate students who were in the lab when I was there. Mario Capecchi is at the University of Utah. He now has his own Nobel Prize. That's Gary Gusson. And I want to tell you about Jim Watson's advice for succeeding in science. Many of you will know that he's quite an eccentric person, but he's also a very honest person. He always calls it exactly like it is. And his first rule is to succeed in science, you have to avoid dumb people. Okay, what does this mean? We all know that if you go out, say you're going out to play tennis, If you play tennis with somebody who's worse than you are, your game probably isn't very good. But if you play tennis with somebody who's better than you are, then you usually play better. And that's what this means. Find the smartest people you can and associate with them. To make a huge success, the scientist has to be prepared to get into deep trouble. And what that means is you have to take on really challenging problems, like Michael Lerner did when he used lupus autoantibodies to discover SNRPs. So you have to be prepared to do something challenging. This is actually about mentors. If you're doing challenging things and things get difficult, it's very important to have friends and mentors who will help you figure out how to get out of those difficult situations. Never do anything that bores you. Well, we all know if you're bored, you're not very interested, you're not doing your best. So you shouldn't bother to do it. And Jim Watson's final rule is that if you don't like to be with people who are also good scientists, who are going to challenge you and question your ideas and question your experiments, that's what it's all about, because somehow everybody has to arrive at the right answer. And so I want to bring this back at the very end to women in science. So why aren't there more women in science? I think you've got the flavor that science is really very social. Jim Watson's rules, all of these are rules about how you interact with other people. So part of the problem of there not being many women in science is that there aren't many women in science. It's harder to make those connections. It's harder to network. It's harder to find mentors. And that's exacerbated the problem. But the problem's gotten a lot better. As I said, you've seen 2 women science professors at least already here today. So what we need to do is encourage people like you guys to do whatever you want to do and to have fun doing it. And if you really enjoy doing science, then you should just forge on ahead, and there will be lots of people to help you to make these discoveries. And just to prove to you that there are some women in science, these showed up at a Halloween party we had a couple of years ago. They're all graduate students. And each one of them is a different SNRP. They're obviously the splice girls. And you can see they're connected by exons. And there's also introns in there that you're not seeing in the picture. So that's what I have to say. And maybe people will come up and do the demonstration. Maybe there'll be questions.